if you are following along my lectures, you must be aware that what is this. Now, if you do not know, and if you do not know how the embryo is formed at this stage, this is the embryo after the folding, after the transverse and craniocaudal folding has occurred. So if you do not understand this, please watch my lecture on development of pharyngeal arches that is available for free on YouTube. So please watch that lecture and this will become very clear. So I'm not gonna explain that how embryo develops up to this stage. You can watch that video. Now the development of ear, especially today's focus will be the internal ear. Development of internal ear starts as a thickening of ectoderm. First let me label these structures for you so that later you won't get confused. So here is the ectoderm. So here is the ectoderm. Here is the neural tube. And then here is the endoderm, and endoderm is basically forming the primitive GI tube, and at this point here is the primitive pharynx that this endoderm is going to form. Before discussing the development of ear, let me tell you briefly that what happens that here there are certain swellings that develop within the neural tube. So three basic swellings develop. Of course there are later more swellings also that arise, but we will not talk about that in this lecture. So three swellings develop. Let me show you. So here you can see that three basic swellings develop, and if I color code them, here you can see these are the three swellings of the neural tube. This is the prosencephalon. Then there is another swelling called mesencephalon. And here the important one which is relevant for us in this lecture is the rhombencephalon. Now rhombencephalon is also called hindbrain swelling, and it is going to form the pons and medulla. At the level of rhombencephalon, we have this ectoderm, and at the level of rhombencephalon, the ectodermal thickening develops bilaterally. This is, by the way, a cross section, and bilaterally there is the development of ectodermal thickening. So here, if you can appreciate, here is the bilateral ectodermal thickening. These thickenings are called placodes, and specifically they are called otic. Otic means something related with ear, so this is otic placoid or placode. I don't know what is the perfect pronunciation for this. And bilaterally here also, this is the otic placoid. Now this otic placoid is going to form almost all of the internal ear, except the outer covering, the otic capsule that we will discuss later. Most of the internal ear, it develops from the otic placoid. This ectodermal thickening, otic placoid, it will invaginate inward. And please remember that they develop at the level of hindbrain or rhombencephalon. So they will invaginate inward within the mesenchyme. Please remember that this is the ectoderm, and let's suppose if I somehow try to peel off this ectoderm somewhat here, let's suppose if I peel off this ectoderm, inside there will be a hole. Of course a hole will be formed here, and inside you will see that it will be filled with a mesenchymal structure which is called mesoderm, or you can say it is a mesenchyme. So this otic placoid, it will grow an invagination, a sac-like invagination into the underlying mesenchyme. So what it will do, it will develop bilaterally, it will develop sac-like invagination into the underlying mesenchyme. To observe this phenomena, we will dive into the embryo like this. So let's go into the embryo. So here is the otic placoid from the inner side. Of course it is filled with the mesenchyme, but we are assuming that we have removed the mesenchyme to observe the structures carefully and nicely. So here is the rhombencephalon, and at the level of rhombencephalon, the Otic placode will form bilaterally sac-like invaginations. Now let's observe those invaginations. So here you can see sac-like invagination bilaterally, right side, and this is the left side. Now this invagination, what will happen to this invagination, that its neck will constrict and ultimately, so here is the neck part, its neck will constrict and it will lose its connection with the ectoderm like this. So here you can see it has lost its connection with the ectoderm, and now it is a separate entity. This ball-like structure or sac-like structure, it is called autocyst. It has several different names. One of the name is autocyst. It is also called auditory vesicle. Let me label it for you. Autocyst or auditory vesicle, and there are several different similar names. Let's not go into that, I don't even remember them. Several authors, they like to put a unique name onto it, but the thing that you must understand, that it arises from the ectoderm, it originates from the ectoderm. This is going to form the internal ear, and its origin is from the ectoderm. Now to understand this further, what I'm gonna do is that I will take you to a 2D diagram and we will see some 2D animations. Let's assume that we have cut a section here. Let's suppose we have cut it like this, and we have cut a section here, and this section, this two-dimensional section, is the thing that we will be seeing in our next presentation. So here is the cut section, and here again this is the neural tube, and basically this is the hindbrain part or rhombencephalon part of the neural tube. This is the endoderm, which is basically the foregut, primitive foregut, and this outer covering is the ectoderm. Now if I move forward, basically there was that formation of Otic placode here. This Otic placode invaginated inward into the underlying mesenchyme. This is basically the repetition. Then this neck will constrict here, and ultimately this connection will be lost here, and now we have this auditory vesicle or autocyst. Now this autocyst, I have changed its color so that it will be easy for you to differentiate from the other structures. Now it will divide into two parts, but before that let me isolate it and let me remove all other distractions. Let's just focus on this. And by the way, please don't lose orientation. Let me first give you the orientation. So basically this is the dorsal side or posterior side, dorsal, and this is the ventral side. This is the dorsal side, this is ventral side, this is right side, and this is left side. Now this is the right side autocyst that we will focus upon, and this is the dorsal side, this is the ventral side or posterior side or anterior side. Please don't get confused about this. Now let's isolate the right autocyst only, and let's remove all other distractions. Now this right autocyst, it will divide into two parts. So here it will divide into two parts, and now these two parts are going to form different components of the internal ear. So here let me tell you that this is again the dorsal side or dorsal side or posterior side, and this is the ventral side. Now this swelling is ventral, and later we will also see it in the 3D model that it is slightly inferior. It is ventral as well as inferior, so it is also called ventral part, ventral swelling or ventral component, or some authors, specifically I'm talking about Larson, Larson also label it as pars inferior. and this is called pars superior, because in 3D model we will see that it is placed slightly superiorly, and also this is placed dorsally, it is near to the dorsal end. So this is dorsal swelling, or it is also called pars superior, and this is pars inferior. And also they have been given the name according to the structures that they are going to form. For example, this is going to form the saccule, so this is also called saccular swelling. Saccular swelling, and this is going to form mainly utricle. Of course there are other structure, but main component is the utricle, so it is also called utricular swelling. Utricular swelling. And by the way, the saccule is also going to form, not saccule, saccular swelling. By the way, the saccular swelling is also going to form the cochlea. So let's see how it forms the cochlea. It's very easy to understand. From the saccular swelling, a diverticulum arises, and this diverticulum, it swells and turns around itself to form specific twisted shape, a snail-like shape, which is the shape of cochlea. So like this, it will form this type of structure, which is the cochlea, and this connection will become very narrow, and this narrow connection that has now been established, it is called ductus reunions. And then this is the utricle part, and from the utricle part, which is also the dorsal swelling, or it is also the pars superior, from this pars superior or dorsal swelling, three disc-like structures arise, and these disc-like structures, they will then form the semicircular canals. Now semicircular canals, how they form, it is not easy to explain on this 2D animations. We will explain it in a great detail in 3D model, but for the sake of completion, just watch this animation, this quick animation that semicircular canals develop. Now we will see how these semicircular canals and how all these things develop in a 3D model as well. Here we have again, here I have marked these things for the sake of orientation. So here we have this otocyst or auditory vesicle. Auditory vesicle is there. This is the anterior side, this is the posterior side. Again, it will divide into two parts, right? So it will divide into two parts, the anterior or ventral part and this dorsal part. And here you can see that this anterior or ventral part, which is also the saccular part, it is, if you focus carefully, if you try to see, it is slightly inferior and it is slightly superior. Although difference is not much remarkable, but it is still there. So this is the pars superior and this is the pars inferior. Now from this saccular part, the cochlea arises like this. I hope you can understand. Let's watch it again. So again, you can see the cochlea develops like this. Basically, it's just an extension from the saccular part. So this extension, this snail-like extension, this swirled extension from this saccular part is basically the cochlea, and here it will constrict to form the ductus reunions. These things you already know. Now, my focus in this 3D model will be the development of semicircular canals. Now, first of all, two disc-like, 3D disc-like structures develop like this. One is facing anteriorly and other is lying at the posterior end, but it is facing on the right side in this 3D model. This is basically going to form this one. I hope you can see. This one is going to form the anterior semicircular canal, and this is going to form the posterior semicircular canal. And this anterior, by the way, it is also called superior semicircular canal. Now initially there is the formation of that disc. Now what will happen, that in the central part of both of these discs, in the central parts, these walls, they become close together and ultimately they will be destroyed. They will undergo the process of apoptosis. Let me show you how. So here you can see, I hope you can see that concavity is going to be formed like this. So right, and on both sides they are almost opposed with each other. Not almost, actually they are opposed with each other, and this opposed wall, it will be denatured, it will be destroyed, it will undergo the process of apoptosis, and now these discs are actually converted into the canals. So here you can see these beautiful semicircular, semi mean half circle, semicircular canals are formed. This is the anterior or superior semicircular canal, this is the posterior semicircular canal, and these two canals, this one of the end, this is connected together like this. So this is the anterior and posterior. Now you must be wondering that we have 3 semicircular canals in the each ear. What about the third one? The third one is the horizontal, or it is also called lateral semicircular canal. What about that? How it develops? Well, first these two develops, then another disc also arise, and through very similar process it is also converted into the semicircular canal. To understand this more clearly, we will cut a section of one of the disc and we will see what happens with it. So here we have a section of the disc. Let's suppose that this was a complete disc like this, and we have cut a section of it. We have removed a section, and then we are observing, and here you can see we have these two walls in the central portion. These two walls, they become close together, they will be opposed with each other like this, and ultimately this opposed part, it will be destroyed. It will be destroyed like this, and a complete semicircular canal is established. So that's how the semicircular canal are formed. And by the way, what is this structure to which these semicircular canals are connecting? And these semicircular canals are connected to this structure just like the uterine tubes are connected to the uterus. And this structure is called utricle. Please remember, it has nothing to do with uterus. Your ears don't have uterus inside them. Uterus is a separate entity, and that is also very important organ, but ears do not have uterus. This is called utricle, just to make you remember that utricle has these connections, has these semicircular canals connected to them, similar to the uterine tube that are connected to uterus. So from utricle and uterus, this sounds similar, so you can remember that. This structure, by the way, is utricle, and to the utricle, these semicircular canals are connected. Now these semicircular canals, one of the ends that will be slightly dilated. They will become slightly dilated like this, and these are called ampulla of semicircular canals. So this is the ampulla, this is also ampulla, and this is also ampulla. Now to understand the orientation of these semicircular canals, imagine that this is a room, and the room, here you can see that this wall of the room, this is the superior wall, or you can say it is also facing anterior. So the interior or superior semicircular canal, it is oriented along the superior or front wall, of the room, and then posterior semicircular canal, it is oriented along the posterior wall of the room, and then there is this inferior or lateral or horizontal wall. So horizontal semicircular canal is oriented along the horizontal plane. Well, to be very precise, these are not perfectly in a precise orientation along the plane. For example, if you focus here, let me show you. This is the horizontal plane. Basically there is about 30-degree angle from the horizontal plane to this, to the plane of this what is this horizontal semicircular canal. And by the way, all these semicircular canals, they are oriented approximately 90 degrees to each other. So for example, if you focus here, this anterior and posterior, they are oriented 90 degrees, and also both of these, they are oriented 90 degrees in relation to the horizontal or lateral semicircular canal. What are their functions? How do they work? We will not go into details in the embryology, but briefly we will talk about that in the upcoming lecture. The details will be discussed in the physiology of the ear. Now all these structures, they will get filled with a special fluid which is high in potassium, that is called endolymph. They will become filled with endolymph, and from where the endolymph will come, again we will not discuss, but just briefly I'm gonna tell you that there is something called stria vascularis. They are within the wall of this membranous labyrinth. By the way, please remember this is called membranous labyrinth. We will discuss more in the upcoming lecture. Now the problem is that the blood vessels, they will be releasing the surrounding blood vessel, they will be releasing the endolymph into these tubes and this network of tubes and sac-like structures. But how these things, how they will be how can they be excreted out? So to solve this problem, another extension from this utricle part will arise, and this extension, this tube-like extension, it will then ultimately here it will form the sac, and this is called endolymphatic duct opening into a sac called endolymphatic sac. So endolymphatic duct and endolymphatic sac, both of these, they have two main functions. We do not know the precise details, but one of the hypotheses is that they have two main functions. One, that they are involved in excretion of extra endolymph. Basically, endolymph is continually being produced and excreted, and the other function is that the sac is actually like a reservoir of endolymph. For example, if endolymph become low from the sac, the endolymph, and from this duct, the endolymph will return back into the membranous labyrinth, so sufficient endolymph may be provided. This is very important structure. If it gets blocked, for example, there is a disease called Meniere disease. So in Meniere disease, this duct gets blocked, and if it gets blocked, there will be problem in both hearing as well as the balance system. So internal ear, it is involved, the cochlear part, it is involved in hearing, and the other remaining part, it is involved in balance. So both of the problems will be there. I will not go into details of Meniere disease, but the basic idea is that it is due to blockade of the endolymphatic duct. Right, so endolymphatic duct is formed, and finally, let's talk about the formation of vestibulocochlear nerves. So basically, how the vestibulocochlear nerve develops, we have these two collection of mesenchymal bodies here. So here one of the collection is this, and the other collection is this. So this cochlear area, in the cochlear area, this collection of mesenchyme, it will divide into certain small parts. From where this come, I will tell you in a moment. Please wait. Let's just assume that they are there. I will tell you from where they come. So this cochlear part, it will divide into certain other smaller section, and they will be arranged in a spiral fashion. These are basically the collection of nerve cell bodies, and the collection of nerve cell bodies, it is called ganglia, and because it is arranged in a spiral fashion around the cochlea, so that is why it is also called, it is not also called, it is actually called the spiral ganglia, or it is also called cochlear ganglia, ganglia of cochlea. So spiral ganglia or cochlear ganglia, it is a collection of cell bodies, and from cell bodies the dendrites as well as axon, they arise like this. This is a bipolar neuron. So dendrite and axon, they arise, and they are going to form the nerve, right? So yeah, thank you so much Imtiaz for your comment. He is saying lovely. Thank you. So here we have dendrite, they are going into the cochlea, and into cochlea to what they are connecting. At this time, these are the empty tubes. Are they connecting to empty tubes? Empty tubes? Well, no. Actually, within the empty tubes, the sensory structures will develop. The organ of corti will develop, and that we will discuss in the next lecture, that how exactly the organ of corti develop. And these axons, they will go outward in the peripheral part, and they will form the cochlear division. They will form the cochlear division of the vestibulocochlear nerve. And what are these two structures? These are not testes. Yeah, I don't know why they are looking like testes, but trust me, they are not testes. Initially, if we go back, it was a single structure, and then they divided into superior, and this is a superior and this is inferior. So they divided into superior and inferior division. Let's draw a diagram here. So here we have the cell body. If I just draw a single one here, I've enlarging it here. So this is a cell body of a single neuron. Now from this single neuron, a dendritic process will arise like this. Dendritic process and axonal process will arise. This is basically a bipolar neuron. At one end, the dendritic process is arising, and at the other end, the axonal process is arising. So here, if you focus, well, let me tell you here first. And here, this dendritic process, it is going into cochlea. Here is the cochlea. So it is going into cochlea, and here there will be the organ of corti. How it looks like, that we will discuss in the upcoming lecture. There is that sensory organ of corti. Here it is connecting to organ of corti, and this axonal part, it will form the cochlear division of vestibulocochlear nerve. And you must be knowing that interiorly there is the rhombencephalon. So between pons and medulla, the rhombencephalon will form pons and medulla, and between pons and medulla, it will connect to that, and between the pons and medulla, there is the nucleus of that vestibulocochlear nerve, and there it will connect with that nucleus. And we will not go into that details, that we will talk hopefully in the neuroanatomy. So yeah. Here you can see that, for example, this is the one cell body. Basically, it's just a collection. You cannot demarcate a single cell body from this. It's not a histological part. So from this cell body, I just focus on one structure. Let me show you. So here is there would be that cell body, for example. And from the cell body, here the axons are going, and here the not axon, this is the dendritic part. Here the dendrite is going, and here the axon is going. So this is dendrite, this is axon, and here is the cell body. So I hope you are very clear about this, right? So yeah, so similar to the cochlear division, the vestibular division of the vestibulocochlear nerve, it also forms. And again, from this, the dendrites and axons, they arise, and the superior part is connecting with the, this is which one? This is the anterior one. You see, it is facing the anterior side. So this is the anterior semicircular canal and transverse or horizontal semicircular canal. So it is connecting, it is taking the sensory information, special sensory information from the anterior semicircular canal, the transverse or horizontal or lateral semicircular canal, and the utricle as well. So this is the utricle. So from these three structures, the superior part of this vestibulocochlear nerve is taking the sensory information, and the inferior, it is taking the sensory information from this one. I think it's labeled wrong. It is taking the sensory information from posterior one. Please correct that. I'm so sorry. It has drawn in a wrong way. It should be here, not here. It's not like this. So yeah, the inferior one is taking the sensory information from posterior semicircular canal, number one, and number two, it is also taking the sensory information from the saccule. So it is also taking the sensory information from the saccule. So inferior one from the posterior semicircular canal as well as the saccule. I think for the embryology, you do not need to go into detail of superior and posterior division. That is important for the anatomy. But anyhow, you have to learn the anatomy as well. So why not now? So these two divisions, they are going to collectively form what we call as vestibulocochlear nerve. So vestibulocochlear nerve basically has two divisions, the cochlear division and vestibular division, and vestibular division is further divided into superior and inferior parts. So this is the situation. By the way, they will go into a canal called canal for vestibulocochlear nerve or vestibulocochlear nerve canal that I am just gonna show you. But before that, let me tell you that from where these mesenchymal condensation from which the nerves arise, from where they come from. This is again a 3D looking cross section, and here you can see this is the neural tube, basically the rhombencephalon part, and here we also have the neural crest cells that are being shown here. So again, here is the rhombencephalon, the neural tube, and neural crest cells are also shown in this diagram, in this 3D model. And again, here is the otocyst or auditory vesicles, and here is the foregut. Now from this auditory vesicles, at the medial wall, from the medial wall of the auditory vesicle, some cells, they separate, they form another layer forms, and let me show you from here. Some cells, they separate from here and they form mesenchymal cells, and what they call is that they delaminate. Laminate means, lamina means layer. So initially these cells were the part of this otocyst, but they separate from this layer, they form a new layer at its medial wall, so they call it delamination. So they delaminate. Basically the exact origin of that mesenchymal cell bodies is not known. So there are three theories for the origin of the nerve cell bodies and vestibulocochlear nerve. One theory is that some mesenchymal cells, they delaminate, they delaminate. So some of the cells, they delaminate from the medial wall of otocyst, and these cells, they are going to form the vestibulocochlear nerve. The other theory is that neural crest cells, they are going to form the vestibulocochlear nerve, and then there is yet another theory, and that I think is the most plausible theory, that there is contribution of vestibulocochlear nerve by both of these type of cells, through this delaminated cell as well as the neural crest cells. So it has dual origin. One hypothesis is that it has dual origin. So three possibilities are there. Either there is the neural crest cells alone that contribute, or there are the cells that delaminate from the otocyst, or there may be the mixture of both of these type of cells. Now in this video, in this 3D lecture, I am gonna assume that the cell bodies, they arise, and of course whole of the nerve, they arise from these delaminated cells. So what will happen that they will divide again into two parts like this, and they will surround the two divisions. This is the pars superior and pars inferior. Pars inferior, of course, is going to form the cochlea. So it will also get divided, this mesenchymal condensation, it will also further get divided into two, and then these two will further, this one will form this is near to the cochlea, the primitive cochlea, or you can say it is near to the saccular division. Not secular, secular is a political view. I mean saccular, sac like secular, not secular. So this is going to form the spiral ganglia or cochlear ganglia, and this will divide into two parts, superior and inferior, and they are going to form the vestibular division or vestibular ganglia. They are going to form the superior and inferior vestibular ganglia, which will form the vestibular division of vestibulocochlear nerve, and this will form the cochlear division of vestibulocochlear nerve that we have already seen previously. And of course everything is going on bilaterally. Again, here we will see that there is the formation of endolymphatic duct and endolymphatic sac. Then what will happen that some of the mesenchyme that is surrounding, of course all these structures are developing within the mesenchyme, so some of the mesenchyme which is immediately surrounding all these structure, basically these are the membranous labyrinth. So all the mesenchyme which is near to this membranous labyrinth, what it will do is that it will condense and it will form a cartilage around it. It will form a cartilage, and this cartilage is like a capsule surrounding the membranous labyrinth, and that is why it is called Otic capsule. So there is the formation of cartilaginous Otic capsule. The central part of the this cartilage, it will die, it will undergo atrophy like this. Let me exactly show you. So like this, it will die, and the cartilaginous part will be replaced with the bone. Cartilaginous part will be replaced with the bone, and here you can see, yeah, so here you can see that now this is okay. So this is the bone, so only this one is the bone, and outside inner to the bone there is this empty cavities, empty cavitation. All this is cavities. Here also cavities surrounded by the bone. Now all these collection of cavities, this is called bony labyrinth. Some people think that this bone surrounding it, this is the bony labyrinth. This is not bony labyrinth. It is still called Otic capsule, but now it is bony Otic capsule. This is Otic capsule, but the cavities, the collection of cavities, this is cavity, all this collection of cavities that we will discuss it in a bit more detail in the upcoming lectures as well. This collection of cavities is called bony labyrinth. So cavities are surrounding the membranous labyrinth. Here if I draw a section of this, you will see that this is the membranous labyrinth. Specifically I am talking about semicircular canal. I will also discuss especially the cochlea in detail, but here is a tube basically. This is the semicircular canal, membranous labyrinth of semicircular canal, and it is surrounded by a cavity. Outside it is the cavity, and that cavity is bounded by a bone called Otic capsule. Semicircular canal, it is filled with endolymph, and bony labyrinth, it is filled with another fluid. Here you can see it is filled with another fluid called perilymph. Perilymph is similar to CSF. It is filled with perilymph. This is the bony labyrinth. And also please note that all this business, everything is basically going on inside the petrous part of temporal bone. So here we have skull, and please note that everything is going on inside the skull. If I hide the parietal bone, here you can see the structure inside. So this is the temporal bone, and how the temporal bone forms, that we will discuss in the development of musculoskeleton system. Let me precisely tell you that this is the temporal bone. Here also this is the temporal bone, and here you can only see one thing, that is the endolymphatic sac here. You know, the brain is covered by three meningeal coverings that are called DAP, dura mater, arachnoid matter, and pia mater. Wait a second. So dura mater, arachnoid matter, and pia mater. So basically it is extradural structure. So here there will be the dura mater, and it is just outside the dura mater like this. Inner to dura mater there will be the arachnoid matter, and arachnoid matter is there, and then inner to arachnoid matter there will be the pia mater, and inner to pia mater there will be the substance of brain. So here there will be the substance of brain. So it is the extradural, this is the extradural structure here, right? This is the endolymphatic sac, which is connected with the endolymphatic duct. Where is the internal ear? Internal ear is basically hidden inside the temporal bone. Make the skull transparent, translucent. Here you can see that the inner ear is situated inside the petrous part of temporal bone. Around this developing inner ear, the mesenchyme will proliferate, and mesenchyme will condense in such a way that it will form the bony framework of the bones of skull. And basically the petrous part of temporal bone, it will be forming around the internal ear. So please don't get confused that what is the precise location of internal ear. That is the topic of anatomy, but I want you to have a crystal clear and three-dimensional concept of anatomy as well as embryology and every subject, physiology, and each and everything I want to make crystal clear for you. So I think that was all about the development of internal ear. Finally, just a quick review that the major thing, the most important thing that you understood, that there is the ectoderm. So there is the thickening of ectoderm. Just a quick review. From this thickening there is invagination, and then this connection is lost here. Here this neck is lost, and this autocyst is formed, and then this autocyst, it divides into, this is the dorsal side, and this is ventral. Now this autocyst, it will be divided into dorsal part, and it will be divided into ventral part. This ventral part is going to form cochlea. It is going to form cochlea and the saccule and cochlea, and dorsal part is going to form semicircular canal and utricle, and the endolymphatic duct and endolymphatic sac, it also arises as a diverticulum from the dorsal part, from the utricular swelling, from the pars superior swelling. All these refer to the same structure. The most important thing that you must know, and that is often asked in MCQs, that which structure is derived from what part. So that is most important. And often people, I know often my students, they forget about the endolymphatic duct, that from where the endolymphatic duct arise. Endolymphatic duct arise from the utricular part. So how can you remember? Please remember, it has nothing to do with urine. Utricle has nothing to do with urine, and it has nothing to do with uterus as well. But for sake of memorization, let's link it with urine. You know, excess water and excess salts, they are released outside the body in the form of urine, right? So U for urine, and here also something similar is going on, that excess endolymphatic fluid, excess endolymph is also secreted, it is also excreted with the help of endolymphatic duct. So this is similar to urine, not similar to urine, I mean functionally similar to urine. This phenomena is functionally similar to formation of urine. So urine, utricle, uterus, they sound similar kind of. Yeah. So in that way you can somehow link these things up, and this will help you memorize this thing. So I hope after watching this lecture your concept will be clear. Thank you so much for watching this video.